Brazil's rich Cretaceous rocks offer a remarkable window into ancient vertebrate history. From a time when South America was still part of Gondwana, the great southern supercontinent. Among the evolutionary stories recorded in these rocks, the origin of snakes remains one of the hardest to decipher. Recently, a paleontological site in São Paulo revealed a new and extraordinary piece of that story. There, researchers found a new fossil snake showing exceptional anatomical detail, a well-ossified adult skeleton including a 3D preserved posterior skull and an articulated vertebral series. Its name, Tametara mirim, comes from the local Tupi-Guarani roots. Tametara means adorned, referring to the sagittal crest, while mirim means small-sized, reflecting the animal's reduced dimensions. In snakes, the skull is one of the most informative parts of the skeleton. Its bones carry signals of evolutionary relationships, but also clues about ecology and lifestyle. The remarkable preservation of Tametara's cranial elements allowed unusually detailed examination of the skull, bringing several key features into view. Among these, separated occipitals, distinct parietal and quadrate processes, together with other cranial and postcranial anatomical traits, support its interpretation as one of the earliest diverging stem snakes. However, Tametara's features were not examined in isolation. Its skull anatomy and fine-scale cranial bone properties were placed within a broader comparative framework, using living snakes and lizards for ecological context, and Deinonychia patagonica as a key fossil stem snake reference, providing a window into the ecological landscape near the origin of snakes. Beyond the skull, Tametara preserves another rare source of information, a natural brain endocast. Formed within the cranial cavity, this structure records the internal space once occupied by the brain, offering an extraordinary view of the neuroanatomy of an early snake. When assessed against living snakes and lizards with different habitat preferences, in contrast with Deinonychia, Tametara's endocast showed a distinctive configuration of specific brain regions indicative of adaptation to subterranean life, corroborating the results of skull bone analyses. Overall, these converging lines of evidence reveal Tametara as more than a remarkably preserved fossil. They show that early snake evolution was not a single path, but rather a complex landscape of anatomical and ecological solutions during one of the most dramatic transformations in vertebrate history.